Welcome back to our flatworm series. Hi, I'm Christopher. In the previous video, we built the cestode operating system. The adult lives in the definitive host, the metacestode develops in the intermediate host, and the exposure connects the two. Now we are bringing that logic into the exam room with one practical question. What was swallowed? Dogs and cats are professional swallowers of suspicious objects. Fleas, rodents, rabbits, raw tissues, fish, crayfish, amphibians, and occasionally something left behind the couch. Cestodes simply take advantage of the habit. In this video, we will organize 3 useful patterns: Dipylidium caninum, the flea-associated tapeworm, the broader Taenia-type prey and tissue pattern, and Hydatidgera taeniformis, the classic cat-rodent connection that may still appear under the older name Taenia-typiformis. Watch for 4 clues: host, exposure, diagnostic stage, and control point. Once those patterns are organized, the species names and distinctions become much easier to retrieve. Let's start with Dipylidium caninum, the classic flea-associated tapeworm of dogs and cats. The adult tapeworm lives in the small intestine of the dog or cat. Gravid proglottids detach and pass in the feces or merge around the perineum. As the segments dry, rupture, or disintegrate, they release egg packets into the environment. Flea larvae ingest those egg packets or the eggs they contain, and a cysticercoid develops as the flea matures. When the dog or cat swallows the infected adult flea during grooming, the cestode completes its journey back to the small intestine. The flea is therefore the intermediate host, not merely a vehicle, because larval development occurs inside of it. The chewing louse, Trichodectes canis, may also serve as a less common intermediate host, but the flea should remain your primary clinical association. The owner may notice fresh or dried segments around the perineum, in feces, or on bedding. We will look more closely at those structures on the next slide. Control must include flea control. Treating the adult tapeworm without addressing the intermediate host leaves the life cycle bridge open and makes reinfection easy. Now pause the life cycle and look at what the tapeworm is actually built from. The adult begins with the scolex, which anchors to the small intestinal mucosa using 4 suckers and a retractable hook-bearing rostellum. Behind the scolex is the neck, the region that produces new proglottids. Together, those proglottids form the strobila, or segmented body of the tapeworm. The younger segments are near the neck and become larger and more developed as they move posteriorly. The name Dipylidium roughly means two-gated, which refers to the paired gated pores, with one opening on each lateral margin of the mature proglottid. Here, the etymology is also a morphology clue. As the segment becomes gravid, it fills with numerous egg packets. The gravid proglottid is not itself an egg packet. It is an entire tapeworm segment containing many packets. When that segment breaks down, the packets are released. Each egg packet contains multiple individual eggs. Within each egg is a hexacanth oncosphere, named for its 6 embryonic hooklets. Fresh proglottids may resemble cucumber or pumpkin seeds. As they dry, they often resemble grains of rice. Egg packets may also be detected microscopically, but routine fecal flotation is not especially sensitive. So keep the layers straight: adult tapeworm, proglottid, egg packet, individual egg, and oncosphere. Tiny structures, very specific packaging. Now, shift from fleas to prey and tissues. This is the Taenia-type pattern. Taenia comes from a Greek word meaning ribbon or band, which is a fair description of the adult tapeworm. The organizing pattern is a carnivore definitive host paired with a prey or livestock intermediate host. Adult tapeworms live in the small intestines of dogs, cats, or other carnivores. Eggs or proglottids pass in the feces. A rodent, rabbit, or livestock species ingests the eggs, and the metacestode develops in its tissues. Depending upon the species, the larval form may be a cysticercus, cianurus, or stroblocercus. The carnivore becomes infected when it eats prey, scavenges, or consumes raw or undercooked tissues containing the metacestode. That is why exposure history matters. Rabbit hunting, rodent predation, access to carcasses, raw offal, or undercooked tissues can move the Taenia-type shelf higher on the differential. A negative fecal result does not erase the life cycle. Additionally, Taenia-type eggs support recognition of a Taenia cestode, but they cannot identify the genus or species by appearance alone. Exposure history narrows the story. Molecular testing may be needed when a species-level answer matters. Hydatidgera taeniiformis is a focused example of the Taenia-type pattern. You may also see it called Taenia taeniiformis in older textbooks and clinical resources. Keeping the current name while mentioning the older one keeps the textbook and lecture from having a custody battle. Here, the cat is the definitive host. The adult tapeworm lives in the feline small intestine, and eggs or proglottids pass in the feces. Rodents ingest the eggs, and the larval stage develops as a stroblocercus, classically in the rodent liver. The cat becomes infected when it eats the rodent. The species name Taeniiformis means Taenia-like in form, which places it neatly beside the broader Taenia pattern. Cat plus rodent hunting suggests Hydatidgera taeniiformis. Clinically, the adult intestinal tapeworm also tells us something about behavior and exposure, not merely what happened to appear in the feces. Now for a diagnostic reality check. Because cestodes are excellent at making us feel confident and then immediately humbling us. Proglottids can be seen in the feces, around the perineum, on the coat, or on bedding. Owners sometimes bring them to the clinic, which is both diagnostically helpful and emotionally memorable. Fresh Dipylidium segments may resemble cucumber or pumpkin seeds, while dried segments resemble rice grains. Taenia-type and Hydatidgera-type proglottids may be broader, opaque, and pass singly or in short chains. Eggs are less cooperative. Taeniid eggs look similar across related genre and species, so they identify a Taenia-type cestode rather than a dependable species. Dipylidium egg packets may be useful when present, but they are not distributed evenly through every fecal sample. Routine fecal flotation can miss cestodes because proglottids and eggs are shed unevenly. A fecal examination is a window into the parasite story, not the entire movie. Use the bigger picture: stage found, host species, exposure history, clinical context, and the diagnostic method. Segments help, egg support, exposure narrows. Treatment, such as praziquantel, removes the tapeworm, but control requires us to break the bridge that brought the parasite to the pet. For Dipylidium, the bridge is the flea. Appropriate cesticidal treatment must be paired with effective flea control. This flea control should be used on the patient, and we also need to consider the environment. Other household pets should be evaluated and treated as indicated. Otherwise, an infected flea can restart the cycle. For Taenia-type cestodes, like Hydatidgera taeniiformis, the bridge is the prey or infected tissue. Control therefore means reducing hunting, scavenging, access to carcass or prey, and ingestion of raw or undercooked tissues when relevant. We may not always convince a cat to stop being a cat, but we can explain the risk, recognize the pattern, and reduce exposure when possible. The tapeworm tells an exposure story, and control works best when we treat both the adult parasite and the life cycle bridge. Treat the tapeworm, then break the bridge. If the bridge stays open, a parasite has a commute. Now let's summarize. Dog or cat, plus flea exposure, plus rice grain proglottids, think Dipylidium caninum. The cysticercoid develops in the flea, the pet swallows the infected flea during grooming, and flea control is part of both the treatment and prevention. Carnivore, plus rodents, rabbits, livestock, scavenging, raw offal, and other infected tissues, think Taenia-type cestode pattern. The adult lives in the carnivore, the metacestode develops in the intermediate host, and infection follows ingestion of infected tissue. Cat plus rodent hunting, think Hydatidgera taeniiformis, formerly Taenia taeniiformis. The rodent carries a strobilocircus in the liver, and the cat becomes infected when it eats the rodent. Diagnostically, proglottids are often the most useful clue. Eggs can support the diagnosis but may not provide a species-level answer. An exposure history helps narrow the pattern. For companion animal cestodes, keep the rule simple. Host plus what was swallowed plus the diagnostic clue equals the likely cestode pattern. Thanks for watching. I'm Christopher Lee, and what you know today protects everyone tomorrow.